My guest today is Professor Joanna Masel, who is Professor of Ecology and Evolutionary Biology at the University of Arizona. She studies foundational questions in evolutionary theory by building models to incorporate mechanistic phenomena within population genetic models and by applying model insights to bioinformatic data. Welcome, Joanna.
Thank you very much.
Yeah, thanks for doing this. So I want to start with your fitness paper. It's entitled Why There Are So Many Definitions of Fitness in Models. And you say here fitness quantifies the ability to survive and reproduce, but it's operationalized in many different ways. Generally, short-term fitness, e.g., expected number of surviving offspring, is assigned to genotypes or phenotypes and used to non-trivially derive long-term operationalizations of fitness, providing insights as to which organismal strategies that evolve due to natural selection. In their essence, you say here, assigned fitness operationalizations are organismal vital rates, births, births, deaths, and organismal growth. So I wondered about this without knowing anything about this. On the surface, these models appear a little simplistic to me. Um, Organismal vital rates, births, deaths, organismal growth. You go on the paper to talk about demographic stochasticity, environmental stochasticity, feedback whereby birth, deaths, and organismal growth cause environmental change. So I'm thinking there's so many variables here, so many uncertainties. How do we tease this out when you look back in time?
So what this paper is doing is its starting point is the practice of modelers like myself. So what we do, biology is really complicated, and we're not going to have total knowledge of any individual system. But we do seek to find general insights. General insights, what types of things tend to evolve? And sometimes it's like, okay, if this type is better than that type, then the better type evolves, the one that's better at being born and better at surviving and so on. And that's kind of trivial. But then there are complicated questions like, does a type that cooperates with its siblings? What are the circumstances under which evolves? Is it better if you are a plant to germinate all of your seeds straight at once, or to keep them in the ground, perhaps dying for several years in order to hedge your bets? What is the optimal rate of bet hedging? Something that will be familiar to your economics audience also appears in biology. And in biology, we have the specific way of formulating it, of asking, what is it that will evolve under natural selection? So modelers basically ask the question, well, imagine there's this and imagine there's that, and let's assume this and let's assume that, and then we derive this insight. Oh, look, this is how bet hedging evolves. This is what will tend to happen. Sometimes weird things happen. There's a phenomenon called evolutionary suicide, where what evolves is eventually really bad for the population. You can get very counterintuitive things evolving. But we set up our models, we gain insight from it. So this paper was sort of noticing the variety of ways that models are set up, that this doesn't entirely align with what philosophers say thought about natural selection and fitness. And we were trying to make sense of the practice of science here.
Yeah, so I had somebody from University of Chicago last year, and he was saying that well, natural selection works. So if you go back to Darwin, so you have some sort of stochastic phenomena going on, different things evolving, and then natural selection happens if you have a better fit with the environment or what is expected of that cohort. He was saying that only works if the genes are not machines in the sense that if the genes can sort of see how things are evolving and programmatically adapt to it, then we are in a sort of a different spot, isn't it? I mean, where do you stand on that idea?
Absolutely. So, I mean, actually goes back to work by JBS Haldane in the, I know it from a book of his in 1932, that sometimes it's advantageous to produce more varied phenotypes, more varied types from the same genotype, and sometimes it's more important what the average is,
Hmm.
and together with very little variation. In a very winner-takes-all system, it's more advantageous to produce a wide range of things because one of them will hit the jackpot,
So you
even if
increase the
that
probability.
costs.
You increase the probability of fit in
That's
some ways.
right.
Yeah.
And it's all about probabilities, but it's not just a single probability, it's the probability of the over the long term that if you have two types, and we call them a resident type and an invader type,
Hmm.
it's about the probability that the invader can take over. And that type comes in, various branches of mathematics, branching process theory in particular, but we have various ways of calculating, will the invader displace the resident?
Hmm.
Will it displace it if it has a better average type, individual? Will it displace it if it has a better extreme, best in the population individual? And that will
Hmm.
depend on the nature of selection. It's not just that you're selecting for one type.
Yeah.
Sometimes you're selecting for bet hedging. Sometimes you're selecting for ability to survive a succession of different things.
Hmm.
Sometimes you're selecting ultimately for the ability to innovate in the future.
Hmm.
And defining fitness in these circumstances, it can be done, is the good news, but
Hmm.
it's not so simplistic.
Yeah. So no, I don't know anything about this, Joanna. I'm just sort of because I'm not a scientist, I can speculate. Um, so if the invader is far removed from the resident, let's say, um, is the probability of a change higher? Well, I think I can answer my own question here. It all depends on what the status quo is, right? And how the environment is evolving. Yeah.
So usually, when the invader is just a mutated version of the resident,
Yeah.
it's usually not going to be that different. But sometimes it might be a whole new species that comes in from another continent and can really displace an existing species. So it's interesting that the same mathematical theory sometimes can apply, whether you're talking about evolution within a species or whether you're talking about ecology between species.
Hmm. So we don't see. Uh, let me ask you: do we see step function changes in evolution? What I mean is that things are sort of going on some sort of linear fashion, and then one day things sort of jump up. It's like the electron um, in a quantum state goes to state one to state two. We don't see that in evolution, do we?
Well, in some sense, nucleotides, they come in G's, C's, A's, and T's. You can't, if you have a C mutating into a T, there is no in-between state.
Yeah.
On the other hand, most of those are very small. I think one of the biggest mutations that tends to happen is polyploidy. So sometimes in a cell, instead of having what used to be one copy of its genome, it ends up with double of everything.
Hmm.
And that can come because two types mated with each other and they ended up with one copy of everything rather than half of everything. Or sometimes within it, it just fails to divide inside the nucleus and you just now get two of everything. And that leads to larger cells and
Hmm.
a whole range of things. And especially in plants, that can sometimes be adaptive. This is a thing, the duplication of the entire genome at once. That's a thing that happens sometimes in evolution.
Mm. So you say here in this paper, population genetic models generally sidestep ultra high dimensional phenotype space and genotype spaces by instead deriving the long-term evolutionary fitness of a lower dimensional set of genetically encoded strategies. Strategies are casually upstream from realized phenotypes. So the conclusion here is that while selection is best understood in terms of differences in organismal vital rates, its derived outcomes are most easily understood as properties of genetic lineages. So could you expand on that a little bit?
Okay, so there's a few things there. One is biology is extraordinarily complicated. There are so many different traits, characters. But at some level, it's simple. We all need to grow, reproduce, survive, reproduce. And natural selection is acting on the fact that we have different abilities to survive, grow, and reproduce.
Hmm.
Uh, so you have this enormous number of dimensions as to what an organism looks like,
Yeah.
and then a relatively small number of dimensions that selection acts directly on. Selection acts directly on births, deaths, and growth. And so the question is, for a biologist, how do we simplify that? How do we put something in between?
Hmm.
How do we say, oh, this is a strategy of bet hedging and waiting for a really good year and counting on that? Or this is
Hmm.
a strategy of tolerance, of eking it out even under difficult conditions. Or this
Yeah.
is a strategy of competitive dominance that only works when there's lots of the same type that crowds something out. And so the essence of biology, in a sense, is to ask the question, how do we make sense out of the enormous diversity? What are the common patterns, the common themes
Hmm.
in the diversity that is biology? Rather than just saying it's all too complicated,
Hmm.
we say, on what axes approximately do the different types use different strategies to try and win? So that's one half of the quote.
Yeah.
And then the other is, what is it that wins? And so there's one view that, it's all about the organism. But to me, that doesn't make sense. If you're about bet hedging, it's not just about the organism. It's about,
Hmm.
a plant that hedges its bets, has lots of seeds, all doing different things. And that plant's long-term success is not
Hmm.
just about the plant, it's about the diversity of what happens to its seeds.
Right.
And then another view is it's just about the gene. And we have a refined version of the gene that we call a lineage view
Yeah.
that I think, we have a better idea of what it means for selection to happen
Hmm.
on genes.
Yeah. So I'm sort of gravitating toward that. I'm thinking selection has to apply on the end outcome. Um, so all the complexities in the multidimensional parameters space um, should not really have an end effect. So if the outcome is the the individual has an incentive to make it highly diversified, so that's sort of a portfolio theory. But then nature itself is going to select out of that 1,000 seeds one of them, and that is based on the outcome. That's based on what that seed is going to do, right? So for me, that makes more sense to me than I don't know anything about this. Yeah.
That's right. Yeah. So we don't see selection as just acting on the organism, the parent. It's acting on the lineage, which includes all 1,000 of those genes being part of the same lineage, and the
Hmm.
fate of the lineage, and that lineage that contains genetic information as to how to diversify.
But do you find any evidence, plants, animals the individual the optimization function would be to create as much variety as possible
Not
when it produce.
always.
No,
Not always. There
yeah,
is such, if you
yeah,
create too much variety, you're sacrificing too many individuals as being poorly adapted. There
yeah,
is an optimal amount, and there's real questions as to what that optimal amount is.
yeah. This is a nice mathematical problem in the sense that it's an optimization problem. And so I
Well,
have some, yeah,
there's a
no,
difference. So
yeah,
I think, for in finance, it's an optimization problem.
yeah,
But it's not just one. You could be optimizing your expected outcome. That's what usually financial people do. But an
yes,
individual who's saving for retirement and investing their retirement portfolio, they might want to optimize for the 90th percentile worst outcome, right? They
mm,
may be risk averse
yeah,
because running out of money is sort of catastrophic. So you
right,
don't want to optimize the average outcome. You want to optimize around the 90th percentile or something, or
yeah,
depending on
yeah,
how risk averse you are, right? So the it's not just one optimization, even in finance. And in biology, the interesting thing is it's not really mathematically, it's not exactly an optimization out process
mm,
at all. It's always a question of which invader can take over which resident. And
mm,
sometimes the answer is that you have two types and each of them can invade the other.
mm,
And in steady state, there is no optimum. There is coexistence between different types,
yeah,
each of which can exploit niches left behind
mm,
by the other. So, and as that fluctuates, everything changes throughout the system, and there isn't really an optimal exactly in the same way.
mm. I don't think I quite understand this. Um, so if I'm a plant and I'm creating seeds, and I say there's so much uncertainty in terms of who's going to survive, who's going to thrive, let's say. And so since I can produce a lot of seeds, I'm going to sort of produce many, many different things on the hope that, some of them are going to be more fit for the future, so to speak. I don't think animals have that luxury because I think, you're sort of programmatically generated. I don't know if plants have the luxury of creating variety at
Oh
will.
no, all organisms have that. So it's not just how many seeds you have, and you've got to remember the more seeds you have, the smaller each one will be, which means that each seed has fewer resources available to establish itself in the world. And so there's a trade-off there.
Yeah.
But the hedging is not just about how many, it's also about when. So
Hmm.
seeds can lie dormant for many years. A seed can germinate the next year. It can lie dormant for a year. It can lie dormant for 2 years, 3 years, 10 years. In
Hmm.
bacteria, there are spores that can lie dormant for thousands of years before they
Hmm.
finally emerge again and grow. So there's also this, it's not just averaging over space, it's also averaging over time. And
Hmm.
animals can do that too. Insects enter a state called diapause, which is a form of dormancy.
Hmm. Yeah. So that makes the problem much more complicated in many ways. And so let me go into another paper, Joanna. So um, order of amino acid recruitment in the genetic code was solved by last universal common ancestors protein domains. So you say here the current consensus order in which amino acids were added to the genetic code is based on potentially biased criteria, such as the absence of sulfur-containing amino acids from the Urey-Miller experiment, which lacked sulfur. More broadly, abiotic abundance might not reflect biotic abundance in the organisms in which the genetic code evolved. Yeah, so um, So let me ask you more of a tactical question. So why did Urey Miller experiment avoid sulfur? What was the rationale?
No rationale, I just didn't add it. I don't think they thought that one through. More to the point, why did people keep using the results of that experiment when there were subsequent experiments that did add sulfur is another. But yeah, so just for your readers who may not know what the genetic code is, I think there's some misconceptions out there that people think that the genome is the code. The genome is the thing that's decoded. So the code is that there are DNA comes in C's, A's, T's, and G's, and it comes in, and basically it gets read as triplets,
Hmm.
CAG, GGA, AAT, in these triplets. And then the code is that there are 64 triplets that
Yeah.
get read, decoded as 20 amino acids. And this is an extraordinarily complex
Hmm.
process. And our question is, how on earth did this evolve? And so we think, well, it didn't just emerge in exactly
Yeah.
its current complicated form. Presumably it started out simpler. In particular, it's probably started out with fewer than 20 amino acids. So we ask, which order, which ones were there in the early codes, which ones were there in the late codes. And instead of just theorizing about it, we
Hmm.
found some methods to get some data from proteins that are so old that they were invented before the genetic code was complete.
So this is pre-LUCA.
That's right. Well, even at LUCA. So we think the LUCA is the last universal common ancestor. It's the ancestor of bacteria together with these other things called archaea, which are also little round blobs,
Hmm.
but they're vastly different to bacteria. And around the time those two groups split off from each other, we think that that's around the time the genetic code got finished, and that the genetic code ultimately was a way of sharing information across life because everyone was decoding DNA in the same way,
Hmm.
and that maybe tree-like life, became more obvious and less of just one big mess after that. So that's sort of as far back as we go. But proteins that are that old or older can
Hmm.
hold clues about which amino acids were early or late, because if something was invented before the genetic code was complete, it will use more of the early amino acids and fewer of the later amino acids.
Yeah. It seems to me that nature was pretty parsimonious. So we have only 4 letters, and the codon is like a three-letter group. Um, as we try to create artificial intelligence, going on a tangent here, Joanna, um, you just put down another layer or create another silicon mountain on the expectation that things are going to improve in AI. But nature was very, very parsimonious here. So this is really a highly optimized system. Do we have any insights into why 4 letters and why the codons are like groups of three?
Ah, good question. uh, I don't think we have solid answer because we don't have, this is the sort of thing we only have one data point for.
Yeah.
um, uh, I'm not saying it's impossible to answer that question. It's just more speculative than I like to get to answer that question.
Yeah, I mean, we don't have any data. I mean, we have one instance of this happening. So it could be by random accident we got there. But even further, 20 or less amino acids going into proteins, that also seems very parsimonious. Perhaps,
Yes. So there are way
yeah,
more than 20 possible amino acids out there.
yeah.
So, these are the 20 amino acids. Well, to be fair, there are actually 22. There are two more that get used occasionally in biology.
Hmm.
One of them mainly in archaea. Um, so, all of life uses 20 amino acids, and occasionally there's two more that pop up here and there. Uh, but there is a much larger set of amino acids that could have been used and aren't. Um, and that is a question, why these 20 plus 2
Hmm.
rather than others, and we don't have the answer to that. But it is contingent. If you were to think, what is life like on some other planet? Maybe
Yeah.
they also came up with DNA, and maybe they also came up with proteins, but it's quite likely that they don't pick exactly the same 20 amino acids as we did.
And it could be environmental constraints. And so one challenge is to, so why don't we have life emerging again today?
Because we'll have to compete with our current life. Yeah, it's
Yeah,
pretty
yeah,
hard. If something's
yeah.
already got 4 billion years to get good at something, it's pretty hard for a newcomer to beat it.
That is true. So what you're saying is that, yeah, so it might be an accident, might have been an accident. So when we go back and look at the accident itself and ask why should it be, there is no real answer to that. I mean, that is how it is. And then we
Yeah,
went
I
from there.
mean,
Yeah, yeah,
natural selection is powerful. Anything that's better at reproducing itself than other things are
yeah,
will take over. And at the very beginning, nothing was very good at reproducing itself. So
yeah.
the bar was lower for success.
Right.
And I actually don't think the origin of life was that hard. I think there were
Mm,
all kinds of chemical reactions happening,
mm,
and some biases existed that certain things tended to be produced and to produce other things in turn. And those biases quickly gave rise to some kind of Darwinian evolution.
mm,
Probably very early, probably in multiple places at once. I think the
yeah.
hard things are things like the genetic code. Really, when do you get really sophisticated, complex molecular machinery?
Yeah. So we had all the raw materials early on for
Yes,
4.5 billion years ago.
yes.
And this happened. Um, but you're saying you're pretty comfortable that given all the raw materials at that time, things will happen. Um,
Yes.
and so the universe then, I mean, we're going on a tangent here, Joanna, just to get your perspective. The universe then teeming with carbon-based life,
I don't
wherever
know the
we
answer
look.
to that. Um, I don't know, it's a little outside my area, and I don't know for sure how many places have all the raw materials.
Hmm. Okay.
We certainly did. What variations in the raw materials were there? I certainly believe the raw materials existed somewhere, but that doesn't mean, just because life existed, doesn't mean that life got as sophisticated as it did on our planet. Life could have stayed much more simple and limited on the
Yeah.
circumstances of other places.
So if you do a more sophisticated Urey Miller experiment again, let's say we put all the raw materials in there, um, what do you think you'll find?
You're not going to find life if that's what you're asking. But you are going to find cysteine and methionine, which are two of the amino acids that were missing because they have sulfur in them. So the real question is, can you get these raw materials or not? So, I don't necessarily, life's not going to appear in one test tube in the space of a few hours. Um, you know, it's very different to say, when I say that it's easy, I mean it's easy on something the size of a planet given some millions of years, which is geologically the blink of an eye.
So you
Uh,
need lot of different experiments, you're saying. So given the variety of experiments that could go on a planet of Earth size, those experiments have different characteristics. They have access to different raw materials. And given the fundamental raw materials that might be there, you're saying that is likely, from a probabilistic perspective, likely to lead to life.
that's right.
But so we looked in the solar system albeit, it's a pretty small system. And every time NASA sends something out, they say, yeah, we're going to find Titan, Acetylus. Yeah, we're going to find life there. Um, my bias has always been, okay, so having a hypothesis is good. So go explore it. But if you don't find it, you have to have some sort of adjustment to your ex ante probability of life. Because if you find all that stuff and say, oh, yeah, yeah, good temperature, good water, flowing water, and all of that stuff, but you don't find any life there, that should tell you something. I mean, it's not that easy. Yeah, at least that's my bias, it seems to me.
So a lot of these places, life is hypothesized to be below the surface, so it's a little bit harder to measure. Um, I know one of my colleagues on the has modeled things with Mars, and it's quite likely that life may have existed there and then burned itself out. So we're going in the wrong year,
Hmm.
perhaps, to
There's
go
a
and
time
find it.
dimension to it. Yeah.
And you have to think that, in most of these cases, the most sophisticated life that we're likely, the best possible case to find is very simple microbial life. I mean, just on Earth, we had only bacteria and archaea for the first couple of billion years.
Yeah. That's a
And
long
then
time.
eukaryotes, which gave complicated cells, of the kind that we have, with something of the sophistication of one of our cells, that's only
Yeah.
about half the length of the history of life on Earth. So if you happened to have hit us in the first half of our history, you
Hmm.
wouldn't have seen anything as complicated as a human cell.
Yeah. So yes, I mean, multiple things here. One is as you say, the time dimension. Things might have happened, might have developed, evolved, and then killed each other with nuclear bombs and went off. So
Not
the
just that, but I mean, I think, for example, in humans, the biggest destroyer of life so far, humans haven't yet reached that achievement. It was cyanobacteria, which evolved the ability to produce oxygen. And oxygen was a toxin that, extremely reactive, that killed most of life on Earth. So the biggest mass extinction event in life's history was probably, in fact, generated by life, but it wasn't humans, it was cyanobacteria. And
that
eventually
bacteria, I
life
never liked
recovered
them.
to it and made oxygen central to how most of life works. But
Hmm.
initially, cyanobacteria produced this extraordinary toxin that eliminated most other life.
Yeah, I mean that's an amazing thing, isn't it? And then that toxic substance gave gave new life, and then developed into something much more interesting. And so, yeah, there are so many different things that need to happen in the system. If you add the probability of all of these things, I think you talk about this in one paper. Just multiply all those probabilities, even if they're 99% probable, and I have thousand different things that need to happen before life actually stands up. The chance of that happening is very, very small, right? I mean, it's so minute that you
I'll
wouldn't
just
think
say
about
we're
it.
not very good at calculating it, because there's also a combinatorial thing that we can
Yeah,
imagine one way of it happening, and there are many other ways of it happening. And so you have to add up all of the different ways something could happen. And I don't
yeah,
think we know enough to perform such calculations reliably.
I agree. So I want to go to another paper, recent paper, the protein domains of vertebrate species in which selection is more effective have greater intrinsic structural disorder. You say here so the nearly neutral theory of molecular evolution posits variation among species in the effectiveness of selection. In an idealized model, the census population size determines both this minimum magnitude of the selection coefficient required for deleterious variants to be reliably purged, and the amount of neutral diversity. At least on the surface, it seems a little counterintuitive, Maurice. So let me read this. The protein domains of vertebrate species in which selection is more effective have greater intrinsic structural disorder. So what does it mean?
Okay, so let's start with protein structural disorder. So most of your listeners probably have an idea of proteins. They've seen some picture with these ribbons in these alpha helices and beta sheets and these well-folded, perfect three-dimensional structures. It turns out that that's not all of proteins.
Hmm.
And many proteins or portions of proteins exist as ensembles of many different structures with long floppy bits essentially and constant movement and no single fixed structure.
Hmm.
And that's known as intrinsic structural disorder. And in species like us, in eukaryotes, it can be a third of all of our protein.
Okay.
So it's not minor. So you might have some idea that, okay, that's some inferior form of protein, and the good form of protein is the well-folded one.
Hmm.
And some agreement with that would be we think of selection being more effective in some species than others. So in population genetics theory, when you have a very large population that can try many things and where statistics average over more stuff, that normally makes selection more effective, more able to get rid of bad stuff,
Hmm.
more able to look at options for good stuff.
So simplistically, so sorry to interrupt, Joanna. So simplistically, can I think about this as the structural disorder gives organism more flexibility?
Who knows? I mean, so structural disorder, so there's a lot of active biochemical research as to what structurally disordered proteins are doing.
Yeah.
Sometimes they actually do fold, but they only fold after they're bound with something else. So they
Hmm.
stay very unfolded until they hit a target, and then together with that target, the binding and the folding are sort of the same thing.
Yeah.
Um, Other times they just seem to be disordered.
Hmm.
And I'd say, you should have, maybe you want somebody on your podcast who's really a biochemistry expert on this, but it is an active field of research. Um,
Hmm.
and then the same protein in different species can be also more disordered or less disordered. It can have longer disordered regions or shorter ones. Uh,
Yeah. So see,
yeah. So,
let's
but if
take
you
a
look at, if you,
yeah.
sorry, go ahead.
No, go ahead. Go ahead. Go ahead. Yeah. Go
So
ahead.
you're saying if you look at bacteria, where
Yeah.
selection's very powerful, they have much less disordered proteins
Hmm.
than eukaryotes like we do. So
That's because eukaryotes have more proteins, or it's just not the number.
that's sort of one of the questions we're
Yeah.
asking is why are there such differences in the amount of intrinsic disorder?
Hmm.
So we came into it as, thinking that basically when you have ineffective selection that's just more tolerant of mess, you'd
Yes.
get more
Yes.
disorder.
Yeah.
That was our hypothesis going in.
Yeah.
And we found the absolute opposite.
Hmm.
So we compared more or less, humans and other primates to rats and mice and rabbits, and rats and mice and rabbits have larger population sizes. Selection should be more effective in them. And
Hmm.
humans and, other primates tend to have much smaller population sizes. Selection should be less effective. We did this, we did a variety of similar techniques, but all along those
Hmm.
lines. And what we found across the board was that basically things like mice and rabbits had more disorder than we do. So this
Hmm.
was absolutely counter to what we were expecting. We
Hmm.
found that this sort of slightly more effective selection increased the amount of disorder rather than decreased it, which was
Hmm.
very surprising to us.
So if I plot disorder to complexity in some way, humans being the most complex, um, as evolution
Oh, I'm
progresses.
not convinced that humans are the most complex. That is, I mean, if you just count complexity as how many genes we have, you know,
Yeah,
humans are not at the maximum. That's a sort of a human bias to assume we are the most complex.
sure. But as evolution progresses,
Ah, I'm not
would
sure
we
that
see,
evolution progresses. Time passes, evolution occurs.
right,
Progress is much more controversial as an idea.
right? So what I was thinking is, as you get more complex animals, let's say, do we find, suppose I sort of go back from human to bacteria and I look at disorder, structural disorder in these organisms, would I find structural disorders sort of declining over time?
Yeah, so this comes back to another, you told me you were going to talk about another of my papers next, and I think I'll jump ahead to that one.
Yeah, yeah, yeah,
So what we found is that the genes that encode for proteins with a lot of disorder,
yeah.
they tend to get lost altogether.
Hmm.
So it's a higher order of selection. So the same gene selection is increasing its disorder. But genes that are very disordered tend to die out completely.
Hmm.
So, you
Hmm.
know, the genes with the staying power to make it for 4 billion years are very, very well folded and with just about no disorder. And the genes that were only invented a million years ago, you know, yesterday in evolutionary terms,
Hmm.
they're often very disordered and their lifetime is shorter. So sort of the disordered proteins are very easy come, easy go in evolution. New proteins are getting invented out of junk DNA all the time.
Hmm.
And they're often disordered and they can replace other disordered proteins and they come and they go. But the ordered ones, you know, so we have these two levels of selection that are going in opposite directions. The classic evolution we think of, it's called descent with modification,
Hmm.
is actually, if anything, pushing the amount of disorder a little bit incrementally up. But things that have too much disorder are getting purged altogether so that they're disappearing entirely.
Yeah. So this is sort of a, I don't know if you think about this an optimization problem without knowing anything about it. I would say so. The disorder is sort of a little bit of flexibility. I could experiment with it. If I have disordered proteins, but then once I find good things, I want to keep them. So then my system sort of becomes more rigid, and so
Yeah, we don't find
yeah.
support for that.
No.
We
Okay.
find support that the things that are very ordered were probably ordered for a very long time already. We don't have a lot of evidence of things going from being less ordered to more ordered. We just
Wow.
see that things that were born more ordered to begin with are more likely to stick around. One
Okay.
exception where flexibility can be useful is sometimes you get, especially in viruses, sometimes you get two different genes
Yeah.
encoded by the same stretch of DNA. You know, I said it was triplets that you read. So you think you could read 123, 123, 123, but you can also read 231, 231, 231, or 312, 312, 312. And then because it's a double helix, there are three more ways of reading it on the other strand as well, reading it in three ways backwards. So sometimes the same stretch of DNA
Yeah.
is read, there are two different genes on the same stretch in different reading frames. And this is especially common in viruses when the genome has to fit into some capsid and be really small. And so there you
Hmm.
actually do see more disorder, partly because that helps it. It has to be more tolerant in order to accommodate what the other protein needs to do.
Wow. Okay. So there are two things going on. If I understand this, obviously I don't understand this. There are two things going on. One is when you find a well structured protein, you tend to keep it. When you don't have that, and then you're sort of battling or struggling for survival or evolution, um, the unstructured part could be useful because you can actually make more interesting things with it. So that's where the flexibility comes in. And so I always, yeah. I think the creationists are going to win here again, Joanna. You can object to the statement. It seems like we have some sort of an end outcome expectation where all the proteins are well structured, well folded, well constructed, and then that's the end outcome for biology, evolution.
But no, it's not the end outcome because new proteins keep getting invented.
Yeah, so the new guys
And
are going to create a wrench into that. Yeah. So, so I just want to touch on a couple more of your papers. Um, Differential retention of PFAM domains contributes to long-term evolutionary trends. So what is PFAM again?
so that's a section of a protein. So proteins sort of they're somewhat modular and they have these domains. PFAM is one of the ways that classification, that are given to the domain. So they're sort of modules. So we find it more convenient to study the modules than to study, if to the pieces rather than studying the whole gene. It's just the whole gene is a bit amorphous. So, but this is what I was telling you about
Yeah, yeah.
the disordered ones being lost. So we don't do that at the level of whole genes. We do that at the level of modular pieces of genes, because those are the things that are modules in evolution, and they're also modules in protein folding.
Yeah, so we talked about this a little bit. So protein domains, you say here, that emerge more recently in evolution have a higher structural disorder and greater clustering of hydrophobic residues along the primary sequence. So there are multiple effects here. So let me go into another one. You've been very productive, Joanna. I got so many of your papers. Extinction vortices are driven more by a shortage of beneficial mutations than by deleterious mutation accumulation. That's also a little bit counterintuitive for me. So habitat loss contributes to extinction risk in multiple ways. Genetically, small populations can face an extinction vortex, a positive feedback loop between declining fitness and declining population size. So that dynamic is well understood, right? So as your population shrinks, you may have inbreeding and all sorts of other issues that may go on that population that makes you less fit, and then there is no way up from there. Um, but you say here there are two distinct extinction vortex mechanisms. Ineffective selection in small populations allows deleterious mutations to fix, driving mutational meltdown, and smaller populations generate fewer beneficial mutations essential for long-term adaptation, a mechanism we term mutational drought. And so at the fundamental level, you say a shortage of beneficial mutations more important than deleterious mutation accumulation, especially in this type of a context, right? When you are sort of in this declining situation, I would imagine the value of a beneficial mutation is a lot higher than anything else. Is that the way to think about it?
Um, Not quite. So it's more about, what is the critical size to avoid one of these vortices? How large does a population have to be in the long term in order to be sustainable? Um, yeah, and the idea was, like we were talking with the proteins before, that selection is more effective in mice than it is in primates. That that's about getting rid of bad mutations.
Hmm.
And so the dominant theory is that when a population is too small, there's a short-term effect with the inbreeding, But even if it survives that short-term effect, in the long term, it just suffers because it can't get rid of the bad mutations.
Mm.
It just, if you like, yeah, they just add up and add up and add up. And what we found is that, yes, that's an effect, but there's this other effect that we think is actually more important, which is that there are just too few opportunities for good mutations to occur. That there's just, with fewer individuals, there's fewer mutations coming along.
Yeah.
And that drought turns out to be at least as important.
Mm. So it's sort of a population size issue.
Yes. Yes. So the paper's all about what's
Yeah,
the minimum size
yeah,
for a population to be long-term sustainable, and why is that the minimum size? To what
right.
degree is that due to the need to purge bad mutations or the need to discover good mutations? And we find that the need to discover good mutations is more
Mm.
important, which was a difference to how it was previously thought.
So I don't know much about this, Joanna, but is our technology advancing to a point that we can induce mutations? What I mean is that if you find a system, a biological system that's going extinct for a variety of reasons, could we go in there and effect a mutation to bring it back?
Well, we're very good at affecting mutations. Radioactivity does it very effectively. But I mean, I think from that perspective, a good place to look is, and this is just, not my area of work, but around Chernobyl,
Yeah, yeah, it'll be a good
you
experiment. Yeah,
there are wild population. Of course, it's been wonderful for local wildlife because the humans don't want to live there, and
yeah,
they have their habitat back, and wild populations are flourishing.
yeah.
As I said, it's not quite my area, but I understand that with all the radioactivity in the soil and so on, the animals become shorter lived,
Mm,
but the populations are completely healthy.
mm.
They just have to shift to somewhat shorter, lifespans, but they might have just as many offspring
Oh,
in those
wow.
shorter lifespan. So,
Yeah.
if you like, we've done that experiment
Yeah.
and the populations are doing just fine with a
Mm,
higher mutation rate. And that's something we've also seen in our simulations,
mm.
that increasing the mutation rate, while presumably being bad for individuals, especially their somatic mutation rate that gives them cancer,
Yeah.
it's actually pretty good for populations.
Very good for population. So that's really interesting. It's also intuitive. And and so the Chernobyl area might give us a lot of data on this.
Yeah, but I don't volunteer to do the research. Not my idea of an ideal field site.
But the intuition is good there, right? So yeah, so the individual suffers from sort of higher level of tactical mutation. It's very, it's not that useful. But then because of the scope of mutations in the population is higher, or the number of mutations that the population is having is higher, the population has a higher chance of not dominating but progressing, right, in a larger volume. Um, but we could do this in bacteria, couldn't we? I mean, couldn't we test this?
Yeah, I'm trying to think what's been done. Bacteria are not the same. So population genetics theory is very different in species that have sex and
Yeah,
species that don't have a lot of sex. And so the results from bacteria don't necessarily translate.
yeah,
But certainly in many bacterial experiments, for what it's worth, and it's not a great comparison, we often do see spontaneously that high mutation rates quite frequently evolve.
Hmm.
in lab conditions.
And so do we see a sort of a negative correlation to lifespan and then a higher positive correlation to the community?
Okay, so that again doesn't translate. I think the correlation with lifespan has a lot to do with being multicellular and having somatic
Okay.
mutation. So I
Yeah.
think we're pushing the comparison to
Too
bacteria too
much.
far.
Okay.
Aging in bacteria is it happens, but it's
Yeah.
I think that's getting too different.
Understand. So I want to finish up with combating SARS-CoV-2 with digital contact tracing and notification, navigating 6 points of failure. And people are saying, yeah, I thought we got over this. Why are we talking about this anymore? And my perspective on this is that the next one, I would argue, is just around the corner. And and that could be even worse than what we've gone through before. I'm a pessimist by nature. So you say here digital contact tracing and notification were initially hailed as promising strategies to combat SARS-CoV-2. However, in most jurisdictions, they did not live up to their promise. To avert a given transmission event, both parties must have adopted the technology. It must detect the contact. The primary case must be properly diagnosed. Notifications must be triggered, and the secondary case must change their behavior to avoid the focal tertiary transmission event. Yeah. So anytime I see a process that I multiply probabilities in sequence, I
Six
know
of them.
that
6 of them in this case.
six of them. I know at the end of that sequence, I'm going to get a very low probability of doing anything. So that's what we found, right? In SARS.
Yes, that's the challenge.
Yeah.
Um, that said, so we wrote this paper. I was very involved in these efforts of fighting COVID. And generally speaking, I am declaring our failure. Obviously, we did not contain the pandemic, but I wanted to write a piece describing what happened so that during the next pandemic, people can learn from us. That said, I think copying whoever did it best
Mm,
last time and building on them rather than whoever spoke loudest is a good idea. And I think of the various efforts I'm aware of for which there's sufficient public data and so on, Singapore did a decent job. And so
yeah.
some of those points of failure, were down to single digits. And when you multiply 95%
Mm,
by six, you still get, 6 times, you still get something acceptable. It's not an impossible task.
mm,
It's just a difficult task that requires political will, coordination, good technological choices, a variety of things. So even though it involves multiplying 6 numbers together,
mm,
that doesn't mean it's impossible. And we look at each of those 6 and we assess the evidence for what values those 6 things had in different
mm,
countries. And it's not a solution that's a priori impossible at all. And I think it's also when you combine a mass testing strategy with digital contact tracing, they
mm,
reinforce each other. So I would not dismiss it. It's not worth putting a lot of effort in, perhaps unless you intend to do it well.
mm,
Half-hearted efforts are not very effective when you have to multiply 6 things to get numbers together. But, although it was a mostly failure this time, I do think there is enough learned
mm.
that it could be possible to do it again. And I think it's also interesting that success breeds success. So
Yeah.
in countries that were very unsuccessful containing COVID, you see declines in trust in government.
Mm.
And in a country like Singapore that was actually extremely successful, you saw increases in trust in government. I
Mm.
think those of us living in Western countries do not see the variety of things that happened across the world.
Yeah, so let me ask you a sort of loaded question. So I grew up in the southern state of India called Kerala,
Mm.
and nearly 100% literacy. They did a really good job. They contained it with all these things that you talk about in the paper. But
I
my
can
question
believe that.
is,
I can
at the
believe
end of
that,
the day,
as opposed to the rest of India. And I don't know a lot about
yeah,
Kerala's situation, but if I had to guess that there was one part of India that contained it, I would immediately guess Kerala.
yeah, but my question is, at the end of the day, so I mean, the next pandemic may not be like this, but um, what I'm finding is that so they really brought the rates down, and then it went up afterward. And so what that tells me is that these really, stringent practical measures, granted, I mean, if you have a hospital capacity problem, that's a good thing to do. But it's unclear to me, in the long run, if that's an optimum thing. So I don't know the characteristics of the next pandemic that is probably around the corner. Uh, if you
If you
get it,
see it,
yeah,
if you see
yeah,
it as waiting for a vaccine, there's enormous benefits to waiting until a vaccine is available before you allow mass infection to take place. So I think, yeah, I'm Australian. I think an interesting case is Western Australia. So Western Australia was doubly protected. Australia was a zero COVID country at quite some cost. Western Australia is highly isolated within Australia, so in many senses it was double buffered. Western Australia, until whatever it was, 2022, had completely normal life with the exception of just a couple of severe 5-day lockdowns. And other than that, bars, restaurants, nightclubs, you name it, life was normal. At
mm,
the end of the pandemic, the incumbent government was re-elected with a completely unprecedented 80% majority. Um, and yes, eventually, when Australia went non-zero COVID, so Kerala was an impossible position because of the degree of connectedness to the rest of India.
yeah.
Western Australia, its degree of connectedness to the rest of Australia was still high enough for it to be problematic. But while Australia was zero COVID, that was manageable. Um, Once the rest of Australia brought it in, which was perhaps a little bit early, but it was approximately the time that vaccines were becoming available, so it was a bit of a race. But for Western Australia, they were able to open up only when people were vaccinated.
Mm.
And
Yeah, so as long
that's
as
powerful. That creates, I mean, the difference in cumulative excess deaths was
yeah,
enormous for doing
right.
that.
Yeah, so as long as there's no test match in Perth, I think you guys are isolated. If you put a cricket game over there, and then everybody is coming from Sydney and Melbourne and everybody else. So,
Well, yes, that's
uh,
what they had. That was the cost. The cost was that they had to cut themselves off.
yes,
But other than that, they could party. Like they were fine.
yeah, that's interesting. So the isolation, so it's two different things going through my mind. One is we have shown that isolation of kids, um, in a very tactical sense, at home, things like that, right, have deleterious effects long term. So shutting down schools and but what you're talking about is sort of a isolation of a geographical area. And Australia has the luxury of doing this because you have a continent. Most countries don't,
That's
right?
right.
So
And then you
uh,
need
yeah,
quarantine. And then you
Yeah.
need a method of containing outbreaks because, of course, no isolation is total. But the point is
Yeah.
that you can, a country that chooses to control COVID, and well, not COVID, but any future pandemic, but a country that chooses to contain a disease can maintain, the question is, what do they need to do?
Yeah.
Obviously, some of what was done with severe lockdowns, nobody wants to do that again. The question is, what could be done that's acceptable, making people who arrive quarantine upon arrival, having certain ideas of testing. In this case, having digital contact tracing so that every time there is an outbreak, it can
Mm.
be brought under control in a matter of days, such that the whole population doesn't, you can avoid putting the population into lockdown. What is it that is necessary to contain outbreaks such that a jurisdiction that decides to control is able to wait for a vaccine
Mm.
and at the same time is able to avoid lockdowns. That's avoiding lockdowns was always the purpose of our, if we did contact tracing and testing well enough, the
Yeah.
purpose of that was always instead of locking down.
Yeah. I just hope we learn from this episode. There's some things that we did wrong. Maybe there are some things that we did right. And anybody who thinks this is not going to happen again is in a fool's heaven. And so the question will be, how do we sort of use the knowledge that we gained to make it better next time? So in that sense,
Yes.
so I
And I
just
think
want
we
to,
should learn from
yeah,
Kerala. We should learn from Japan. We should learn from Singapore. We should learn from Vietnam. There are, I think, there is a very Western bias to narratives.
but these are small systems. So,
Japan is not
if
small.
sorry,
Yeah, Vietnam is not small. Japan is not small.
yeah, Japan is not small, yeah, but you get a population like the Japanese, you can do pretty much anything you want. Uh,
Yes, but they
so
chose the right things. Like if you choose
yeah,
to focus on hand washing and the pandemic, the disease is airborne, it doesn't matter that you get huge compliance with hand washing. It's not very helpful. I think the important thing is the Japanese declared that it was airborne in February 2020. They followed, unlike everyone else, they followed the evidence. They did not lock down. They wore masks. They put
yeah,
carbon dioxide monitors everywhere. They improved ventilation. They allowed work from home, which was an enormous
mm,
change to Japanese culture.
yeah.
And they
So the
kept the schools open and everything else going. I mean,
yeah,
they made, I think Japan made some excellent choices.
yes, yeah. I mean, it's going back to the evolution evolution question here. If you have a fairly uniform system like Japan or Singapore or Kerala, it's very
Singapore
easy.
is not, I mean, it's
Singapore is
governmentally
not.
uniform. It's
Singapore is not.
racially and
Yeah,
culturally
but
diverse, but
it's small in the
it's
grand scheme
it's
of
it's
things. Yeah,
small. It has a single government that is city, state, and country. So
yeah,
unified government, somewhat on the autocratic, benevolent autocracy
yes.
variety.
Yeah, so we say so. But in large systems, like the US, in India more generally, where you got India has 1.4 billion, China has 1.4 billion, US has 330 million. These large systems, highly diverse. And I mean, China is a good example of a large system driven by autocratic rules. Um, Yeah, it will be interesting to, yeah, but I didn't see significant positive effects there either. I don't
So
know.
China was
Yeah,
such a tragedy. I mean, I think
yeah.
Western coverage of China, I think, was very poor, but China had a zero COVID policy that allowed people forget how much time the average Chinese spent in normal life while
Hmm.
Westerners were locked down. They only remember the the post-Omicron failed, ever escalating lockdowns. But I think it varied a lot where you were in China. If you were near the North Korean border, you were in, you know, almost constant lockdown because it kept coming over the border from North Korea
Oh,
again and
yeah.
again and again. Whereas if you were in other parts of China, if you were sort of far from the big hubs with lots of migration, you were more like Western Australia. You were living completely
Hmm.
normal life for the vast majority of the time.
Hmm.
The tragedy of China is that they did not accept that their vaccine wasn't as good and import foreign vaccines. And when
Hmm.
they finally let COVID rip, they had an ineffectively vaccinated population and huge excess deaths that did not occur in places like Australia, who had received more effective vaccines.
Hmm.
And that was just a real self inflicted
Yeah,
insanity that happened in China, that they just wouldn't admit their vaccine, own homegrown vaccine was less effective.
yeah. So I'll finish up. I know that I have to let you go. So, um, uniformity. So Japan, China included, in a discontinuity, you can implement policies, isolation policies, Western Australia in some sense. Isolation policies are implementable, but in
They were
diverse,
implemented
large,
in Australia as a whole, and Australia is an individualistic Western country. It had an
I meant
advantage of
Western
for
Australia, where you had the geographical,
yeah, but all of
um,
Australia, all of
yeah,
Australia, all of Australia embarked on zero COVID. Western Australia had it
hmm,
nearly cost free. You know,
yeah,
Victoria and New South Wales, especially Victoria, bore most of the cost.
yeah,
But all of Australia consented. Well, not all the individuals consented, but a individualistic Western society that's culturally diverse also, with geographic advantages. I mean, I
hmm,
don't think this. I don't think that some of these narratives fit so much. There's this narrative that only sort of autocratic, homogeneous populations could do it, and
hmm.
I don't think that's factually correct.
Yeah, I mean, it's interesting. And so Scandinavia, we think sort of uniform. Sweden, Finland, they had a different policy. They basically said no isolation at all.
Oh no, they
Sweden,
had
for
very,
instance.
very different. No, no, hang on. Sweden
Yeah,
was different to the rest of Scandinavia.
yeah.
So, and you can see it in the excess deaths policy. So, Sweden's excess deaths are substantially higher than other Scandinavian policies' excess deaths.
Hmm. Substantially higher, but in the long run. So there are two metrics we have to measure, I think. One is sort of the tactical impact of the pandemic, but then you have to also measure so the long-term effect. I mean, this is why
Yeah,
I'm, yeah,
cumulative excess deaths
yeah, cumulative excess death. But for how long a horizon is the question? Yeah.
until the end of pandemic measures, right? And cumulative
Yeah.
excess deaths, Sweden turned out substantially worse than its Scandinavian neighbors.
Hmm. Okay. Yeah, yeah, I should look at this data. Yeah. So I was under the impression that Sweden did okay. But then, Sweden has a small population. So if you say cumulative excess deaths divided by the population, that ratio might be quite is high. Then the policy hasn't been effective.
That's right.
But I
Yeah,
don't know.
XX deaths are sort of a fancy thing based on how many you normally expect, given the size and demographic makeup and so on of the population.
Yeah. So now we have a lot of data. Let's get ready for the next one.
Yeah, and I think learn
Yeah.
from the countries that, and a good metric of success
Yes.
is who had low cumulative excess deaths. Like that seems to me
As a
one
percentage of the
metric.
population, I would say, right? As a
Yeah,
percentage of the
that's what I mean.
population.
Per
Yeah.
capita, yes.
Yeah. Excellent, Joanna. Yeah, thanks so much for spending time with me.
Okay, thank you very much for having me.